name is Sahara, and I'll be explaining the benefits of biofortification of calcium to treat osteoporosis. So the solution to prevent osteoporosis is to increase the amount of calcium absorbed in one's diet. Calcium is very important because it helps with having strong. It helps us to have stronger bones and teeth, and it also increases the amount of osteoblast cells in our body. Osteoblast cells, they're important in formation of new bones because every 6 to 8 months new bones are formed, and we need those osteoblast cells to contribute to this formation, and so we can have new bones formed. In fact, there are many people who rely on plant diet and they cannot use dairy products because it's harmful for the environment, and there are also people who cannot use calcium pills. So therefore, in order to fix this problem, there have been problems like this. There have been a lot of attempts made through using biofortification, such as fortifying rice with vitamin A that would prevent blindness. So we will be fortifying potatoes to prevent osteoporosis and to increase the calcium absorption in our diet.
Hi, my name is Jimenez Trejo, and I'm going to be informing you guys about the ethical consideration of biofortified calcium into potatoes. Now, one of the problems that causes or should rise is this is a genetically modified plant, and not that many people are openly to GMOs while others are. The people that are not openly to GMOs believe that this could be a harmful health risk and also probably an environmental risk. However, if we do want to start this and it does get accepted, we will need 3 approvals of the United States Department, which is the United States Food and Drug Administration, the Environmental Protection Agency, and the United States of Agriculture. Once those approve and believe that this could be an important addition to our food source, we could start trials with mouse experiments, and if the data shows beneficials, we could send another application to FDA where they could review it and accept it and allow us to do human trials. Now, another part is if this does get accepted to the human food market, we will need proper labeling on these GMOs potatoes, because there are some individuals that may try to take these that do not require the extra calcium. And with this, if any individuals take these extra calciums, they could have an increase of hypercalcemia or organ calcification. Hypercalcemia is when there is a high increase of calcium in the bloodstream of the individual that could actually cause tumors, while organ calcification is when there's a high enough of calcium causing the organs to harden. Another reason that we will need is vitamin D supplementation, because in order for calcium to be absorbed, it needs vitamin D. And with this vitamin D supplementation, it could cause an increased absorption of calcium in the digestive tract.
Hi, I'm Lauren, and I'm going to be talking about osteoporosis. It's one of the world's most prevalent nutritional diseases. To put it in perspective, 1 out of 2 women over the age of 50 are going to be diagnosed, and 1 out of 5 men over the age of 50 are going to be diagnosed. This is caused when the body loses too much bone density, or in other words, can't produce enough bone. This is a representative of what it would look like under the microscope in comparison to a healthy bone. As you can visually see, their spaces are way larger here, which causes a decrease in the overall density and mass of the bone, and in turn causes these brittle bones that often break. The results could be awful and can lead to disabled mobility, loss in height, and hunched posture. This disease usually hits towards the hip, the spine, or the wrist. And for example, how badly it could get, 20% of seniors who break their hip will die within a year.
Hi, my name is Latham, and I'm going to be talking about the goal of our project. Considering osteoporosis is such a prevalent disease in the US and the world, we wanted to find a way that people can get more calcium in their diet without relying so heavily on the dairy industry. We found that biofortification efforts in the past had been successful, such as golden rice fortified with vitamin A to fight blindness, and flour fortified with folic acid. So we wanted to do something similar. We decided to seek out one of the most widely available and widely enjoyable vegetables in the world, which happens to be the potato. And of all the potatoes in the world, the most popular is the russet potato, also known as Leonard tuberosum. Our goal was to extract the X-Cax gene from a thallocress plant, also known as Arabidopsis thaliana, and insert it into the russet potato. This gene is a calcium exchange transporter and helps the body absorb more calcium from foods by exchanging calcium and potassium ions for sodium ions. Our hope was to boost calcium absorption overall to help diseases like osteoporosis.
My name is Michael Gonzalez, and I'm going to be discussing the methods of our experiment. In particular, I'm going to be talking about the extraction of our transgene and how it's going to be expressed in the potato. X-Cax1 will be extracted from Arabidopsis thaliana, also known as mouse ear cress, by lysing the cells of the plant. PCR will then be utilized to amplify the gene. After many copies of the gene have been manufactured using PCR, they will be inserted into our first of two plasmids. X-Cax1, when it's strongly expressed, it significantly increases the readily available calcium in the edible portions of plants. The way that X-Cax1 works is by encoding potassium-dependent sodium-calcium exchanger proteins, and they work by exchanging a calcium and potassium cation in exchange for 4 sodium cations. Since this experiment is a plant-to-plant transmutation, splicing of introns will not be necessary. The promoter that we're going to be using is CANV, and the reason why this promoter is going to be useful for us is that mouse ear cress and potatoes are dicot plants, and CANV is a strong promoter of dicots, but is not a particularly strong promoter of monocots.
Hello, my name is Peter, and I'm going to be talking about the plasmids. So first, we need to make sure that the plasmid is going to be taken up by the bacteria, which is then going to be introduced into the potato. And the way we do that is we place the plasmid and some bacteria, which we hope will recombine onto an agar plate, and then put some ampicillin on there. Now, the way we make sure that the plasmid enter the bacteria is by putting ampicillin, because there's an ampicillin resistance gene in our plasmid, and the bacteria that didn't take up our plasmid will just end up dying. And we will know that all the ones left have taken up our plasmid and therefore have our transgene. Since our project is basically done through consumption and the potatoes will be eaten up by us, we need to make sure to remove the ampicillin resistance gene, because if it stays in, then when the food goes down through our intestines, it will lyse, and then the bacteria will take up this ampicillin resistance gene, and then you will have ampicillin resistant bacteria. Anyway, the way we remove the gene, or the neo-R, the ampicillin resistance, is by mating 2 different organisms, one with the plasmid of our transgene and the other with Cre. And what will happen is the Cre will attach to the LOX-P sites, kind of cutting out, not cutting, but removing the neo-R from the plasmid with our transgene. And then we will just have the organism, which is our potato, with a plasmid with only a transgene.
Hi, my name is Leonard Tabora, and I'll be talking about the methods of our project. In particular, I'll be talking about recombination and how it will be used to genetically modify a potato. Our potato is used so that we can introduce a X-Cax gene, in which would be consumed by people that have a problem with osteoporosis, people that are calcium deficient. Now, what we need to use for this potato and introduce it is argobacterium. Now, what's special about this bacteria is that it's very, very efficient in infecting DNA especially plant DNA, and so it's used to introduce our transgene. Now this is done by using the Korean lock system. As you can see, we have 2 plasmids of containing an argobacterium. Now what's introduced in these plasmids are a transgene, LOXP, and a resistance gene. And then we have another one over here containing a Cree gene. From there, we would have to infect a potato with our argobacterium. They will then intake the plasmid and contain whatever was in the plasmid from the argobacterium. And same over here, Cree gene infecting a potato containing a Cree gene. This will ultimately create our parent generation. And in order to get our desired SCAX gene alone, we would have to mate them by using the Korean LOXP system. Now, how it works is that whatever gene is in between the LOXP site is used to recognize, Cree is used to recognize wherever LOXP is and splice out those whatever in between. This is used to get rid of the transgene or the resistance gene, and because it's not safe for consumption for use, and so it will make our potato an XCAX gene for F1 generation. From there, it will be ready to be breeded and used for people that have osteoporosis and used for calcium deficient people. Thank you.